All right. Well, thank you for being here, and thanks for the invitation. I really appreciate the opportunity to come over and discuss some of what my group has been working on. In trying to put this talk together, wanted to sort of step back and take a very large, wide-angle view of genomics and how hereditary conditions might work, and then focus in with a couple of examples from work that my group has been involved in. So for the very basic slides, I apologize to those of you, but I assure you that we will catch up very quickly and get into the meat of what I'm talking about. So I just want to emphasize at the outset what the importance is in genetics of having a diagnosis. And for those of you who are clinicians, you understand this already. Regardless of whether a genomic diagnosis can change someone's management, having a better understanding of that patient's diagnosis and what is causing their phenotypes can give you a better understanding of their natural history, predict their course, help the family understand who else in the family might be at risk, how did this happen, transition from a diagnostic odyssey to a management plan. And so these are all very critical aspects of genomic diagnosis. I think certainly on the US side of things, we're facing difficulty with having insurance companies reimburse for genomic tests, understanding that a diagnosis in itself is important, and that's something that we're struggling with. And I think that our field needs to really emphasize that this is really the central job of genetic testing. Many of you are familiar with this slide. In fact, Dr. Rubin showed an example of this, but just to sort of emphasize that genetic variation in humans comes at a variety of different frequencies, ranging from common to rare, and those different variants probably have different effect sizes in terms of the phenotypes that they're impacting. And primarily for the genomic rare diseases, we're talking about the rare alleles that cause high penetrant conditions, versus the common alleles that cause multifactorial conditions. Another way to think about that is to place genomics in the context of the environment. And so here I'm showing the genetic influences at the bottom, essentially your genetic variation that you start your life with. And those include a combination of common alleles that put you at a low risk for certain health conditions, a combination of high-risk monogenic alleles, many of which may represent carrier status, but some of which may be disease-causing, and then a variety of moderate-risk intermediate alleles. And then on top of that, we're going to layer all of the events that happen to us throughout our lives, chance relating to the radiation that our bodies receive daily, the natural effects of aging, environmental impacts like toxins, carcinogens, and our behavioral things that we do to ourselves, diet, exercise, smoking. And if we put this together, just to show you this sort of graphically, imagine a cup that's being filled on a daily basis with all of those non-genetic influences, like a cup being filled with water, and your genetic influences being like small marbles in the bottom of the cup. And you can see that for conditions that are multifactorial, you may have a small number of common low-risk alleles, and it may take a long time for that cup to fill up and for the disease to manifest. Compare that to a monogenic condition in which there's a very large marble that fills most of the cup. It doesn't take very much time or random chance for you to develop that condition. And of course, in the middle, we have the more complicated scenarios. We have individuals who have a higher burden of common multifactorial risk SNPs that may have a higher risk for developing disease in their lifetime. And then something that I think our field is going to be increasingly facing as we do more genomic sequencing and discover rarer alleles with a moderate impact is sort of an oligogenic effect of both small effect size and moderate effect size alleles. So I want us to think about this entire spectrum of disease etiology, even though for the rest of the talk I will primarily be focusing on the monogenic high-risk alleles. Some other considerations that we want to take into account for monogenic disorders are the types of variation that can cause those conditions, and it's a very broad range from small single nucleotide variants to large copy number variants and so forth. The origin of those alterations can be inherited, they can be de novo, they can be somatic mutations that happen, such as in cancer, but also in some of the brain disorders that you'll hear about later. We need to think about what is the cellular and organismal effect of those variants. Are they resulting in a haploinsufficiency mechanism disease, loss of function, dominant negative, gain of function, et cetera? Whether they require 2 alleles to be mutated or 1 in terms of the zygosity, the penetrance of those conditions and the modifiers of penetrance, the expressivity of those conditions, and even other unusual mechanisms that are also relevant. So these are all things that are affecting our ability to make a genomic diagnosis that need to be taken into account. The other thing that I would just like to emphasize is the genetic variation size varies over many log orders from a single nucleotide variant to an entire chromosome, and that our technology needs to be able to discover all of these variations throughout the entire size range. So just to put that into perspective of evolving technology, you know, in the good old days, we had pedigrees and we had markers across the chromosomes that could be used for linkage analysis, and that was not very high resolution. We had karyotypes that allowed us to count chromosomes or look for large translocations. And technology has increasingly been taking us into the realm of discovering smaller and more specific variations, ranging from chromosomal microarray to next-generation sequencing, and even now some of the microfluidic methods of detecting structural rearrangements. And so I think, you know, the take-home here is that genomic analysis does need to reliably detect all types and sizes of genomic variation. And it's certainly driving us towards a unified approach, but we need to be aware of our blind spots. So what are some of the benefits of genomic sequencing in this context? So clearly for conditions in which there's a great deal of genetic heterogeneity, intellectual disability being a great example, with hundreds, potentially thousands of different genes may play a role in that condition. Certainly, by doing genomic sequencing in patients with rare disorders, we can better characterize the phenotypic spectrum of those conditions. Where currently much of what we know about these conditions comes from small numbers of case reports in the literature, we need to start amassing great databases full of characterizations of patients with these rare conditions so that we can give our patients and their families better information about natural history. We do need to have a mechanism to catalog that phenotypic data and better ways of sharing data among different communities. Certainly, genomic sequencing has proven to be a fruitful method for discovering novel etiologies for disease, trio sequencing, discovering new de novo mutations, and so forth has been an incredibly productive area for our field in the last few years. I will point out that the diagnostic yield of any testing is going to depend on the assay. Trios have been better for detecting certain types of mutations, but in fact, exomes and genomes have different capabilities, and we need to keep that in mind. So just as a basic primer on clinical interpretation of genetic variants for those who don't routinely do this, I want to point out that we have to be very cautious in the clinical realm about two aspects of this. The first being the gene-disease association. And how confident are we that there's a clinically valid association between a gene and disease? And this may seem like a fairly straightforward problem, but in fact, in rare disorders where there may only be a few people in the world described in the literature with a particular variation in a particular gene, we do need to be cautious about how we interpret that data. Secondly, with genetic variants, we understand that clinically speaking, there are, in the US at least, 5 categorizations that we use to describe variants, ranging from benign to pathogenic. And this effectively means that there's a huge range in the middle of variants of uncertain clinical significance. And we use those clinically, or at least we expect them to be reported clinically, because they may be of some value if we are able to perform family segregation studies or just to learn more about the variant over time. And so they can be possibly informative to have these variants of uncertain significance, but they also create many issues in terms of the counseling of those patients who have uncertain variants, and they can also lead to potentially false positive results. So Clearly, clinical genome scale analysis is now widely utilized for diagnosis of monogenic conditions. I will point out that this is a complex medical test, and I think it requires careful thought and counseling of patients before it can be applied in a rational way. There have been a number of really comprehensive articles. These are two from JAMA from a couple of years ago that really provided the experience of two large clinical labs in exome sequencing in the US. And effectively what this tells us is that the diagnostic yield of exome sequencing is roughly 25%, probably higher when you look at different conditions. Of course, the diagnostic rate is going to depend on whether patients have already had a previous evaluation or whether they are essentially naive to genetic testing. It'll also depend on the type of condition, where some conditions are going to have a higher yield than others, for some reasons being that we know more about the genetic variation that causes certain ranges of conditions, or we know more of the genes that are involved. So some of the challenges of genomic sequencing in the clinical perspective, I broke them down into two things, one being uncertainty and causality. So our job here with genomic sequencing is to assign causality to a specific genomic variant. And part of that is understanding for a given patient, when is that genetic finding a good fit for their phenotype? And this is requiring us to understand the phenotypic spectrum as we understand it from the literature or from other known patients. It also requires us to think about the variant and whether that variant is pathogenic or uncertain. And we need to think about how we're going to deal with the rapid evidence accumulation around genes of uncertain significance, which may only have one or two known examples in today, but tomorrow or a week or a month from now may have a dozen other examples of patients with apparently pathogenic mutations in those alleles. So these are things that are challenges to the field. I think many of the sessions that I've seen at this meeting actually will address a number of these. I think it's going to be a really interesting time for us. The other thing that I'd like to focus on, and I'll talk about some of my group's experience with, is secondary findings and thinking about what an actionable finding is or a non-actionable finding is and how that might relate to informed consent and preference setting. So I've been involved in a study called NC Genes, which looked at exome sequencing of patients with suspected genetic conditions. We analyzed about 650 patients and, like many other studies, found that diagnostic yield varied greatly based on the condition. I won't go into the details of that. One of the major things we addressed was how to handle secondary findings. And very early on, our group identified what we called 3 bins of genetic information, one being all loci that had what might be considered to be clinical utility or high clinical actionability. And these would be findings that if you identified them in a person without that disease, would strongly predict the development of a condition that could be preventable or treatable if caught early. Bin 2 being loci with clinical validity, meaning that they are clearly associated with disease but may not give you a targetable, preventable condition, but might, by disclosing that information, lead to psychosocial harm. So the question for us was how to draw the line between bin 1 and bin 2, and we did this by defining a metric for actionability in which we sort of came up with these 5 criteria, one being the severity of the disease, 2 being the likelihood of the severe outcome happening in an individual, the 3rd being effectiveness of the interventions that you could apply, the 4th being the acceptability of those interventions to a person who did not yet have disease, and 5 being the knowledge base. And we just simply scored these on a 0 to 3 scale and tabulated them up. And this gave us a relatively rough way to assign actionability. Of course, there are many caveats to how we did this. But we applied this initially in 372 gene disease pairs. We've now gone up over 750. The important thing to take from this is that we looked at some genes that we had previously thought would be in bin 1, as well as genes that the American College of Medical Genetics thought were medically actionable, as well as a subset of about 100 and so on random genes. And the scores from those groups indicated to us that our bin 1 genes that we thought were actionable, as well as the ACMG genes, both scored at a very high level, whereas the random genes predictably gave us a much wider spread of scores. And that told us that our metric was probably differentiating between genes, or between the conditions. And the top quintile of the random genes was 11, and so that's where we have sort of drawn our line for right now. In NC genes, we analyzed all the BIN1 genes in our participants and had about 3.5% with positive findings, and that's pretty consistent with what other groups have described in terms of positive secondary findings for actionable conditions. We also randomized the adult participants in the study into a trial of decision-making about additional information. And we divided up the BIN2 conditions as shown here: multifactorial GWAS risk SNPs, pharmacogenomics, carrier status, ApoE status, other Mendelian disorders, and then severe neurological disorders. And we simply asked people to make a decision. We had 155 participants that were eligible to request those categories, and we provided them with information about those conditions and asked them to go home and think about it and then give us a call and let us know what information they wanted. And when leaving the clinic visit, about 3/4 said that they would probably request at least some of the categories. But in fact, only 1/3 actually called to request that information. So there's a differential between people's intentions and people's actions. And I think this is really important to take into account when we think about all the survey research that's been done asking people if they would like to know their genomic information. Far fewer actually took us up on the information. When they did, however, they didn't really differentiate between the types of information, so the vast majority chose all types of information. And I would point out that this presented additional challenges from the genetic counseling side, from the genomic analysis side, and really created a lot of extra work for very little in the way of important clinical findings. And so I think this raises some important points about whether this is something that we should be routinely doing in clinical care. I will just point out that the NC Genes team was a very large group led by Jim Evans at UNC and had a large number of participants, and I don't have time to go into the details there. So let me switch now to genomic screening for the remainder of the talk. And I want to point out that, again, as we found from the incidental or secondary finding studies, about 3% of us have rare genetic conditions that are preventable or treatable if diagnosed before symptoms develop. And we all, of course, hope that genomic medicine can be proactive instead of reactive. We don't want to wait for people to develop disease if we can identify it and prevent it from happening. However, it is important to note that there's really a great deal that we have yet to understand before we can really implement this widely in an evidence-based manner. And here I think context is key. When we're thinking about the diagnosis or prognosis or management of a baby, of a person with a suspected genetic condition, for example, a sick baby in the neonatal intensive care unit, this implies that there's a disease process present, or at least some prior probability that they might have a genetic condition. And we use our molecular tools to dissect that pathophysiology to provide an etiology and impact their outcomes. In contrast, if we're thinking about the screening of a healthy infant, the individual themselves has a prior probability that's that of the general population. So the prevalence of that condition defines that person's prior probability to have a gene alteration. And really, we're predicting the likelihood of developing disease in the future. And for those that have studied clinical epidemiology will know that the very low prior probability of these individuals having disease means that any imperfection in our genetic testing, meaning less than 99.9% specificity, will result in large numbers of false positives. So one of the challenges here is that when we think about traditional newborn screening using biochemical markers, those Analyte tests can be very clearly calibrated based on what is normal in the population. So what I'm showing here is the sort of theoretical Analyte concentration for a given biochemical marker, where the unaffected population has a very nice peak at one particular level, and the affected population has also a very nice peak, and these are very well separated from each other. And if we zoom into the small area of overlap, you can see that one could set a threshold at one side where we would minimize the false positives by essentially setting that threshold just past where the unaffected population range is, but we would have some false negatives at that particular threshold. Similarly, if we were to set it at the other end where we minimized our false negatives so that we would catch all of the affecteds, we're then introducing many false positives. And in newborn screening, we're able to actually calibrate that to be able to achieve the desired ratio of false positives to false negatives. The question I raise is there an equivalent mechanism to define what is a positive screen for genetic variants? We've been addressing this question in the NC NEXUS project, in which we have 2 cohorts, one of which is healthy newborns, and we're enrolling the parents prenatally, so we've identified them through the healthy obstetric clinics, as well as a cohort of infants and children where we already know that they have conditions that are identified through newborn screening. We used the semi-quantitative metric that I showed you previously to define categories of information that we would return, and I'll show you that on the next slide. And the parents completed an online decision aid prior to having sequencing so that they could understand what the exome sequencing would be about and what kinds of information they might learn. In our study, all the parents have received results, if any, for the childhood onset medically actionable conditions that we called the next generation sequencing newborn screen. And we then randomized the parents again into a decision arm and a control arm for additional genomic information. So in this sort of diagram, what I'm showing here is the semi-quantitative metric score that I described for you from 0 to 15, now with the x-axis being the age of onset or the age of intervention. And essentially what we said is that there's basically 4 quadrants, and if we took 18 as an arbitrary designator of an adult, and I will admit that that is an arbitrary definition, things that are having onset before age 18 would be considered childhood onset, and those with higher actionability would be considered part of the next generation sequencing newborn screen. Whereas things with lower actionability would be part of a parental decision group, essentially pediatric onset, non-medically actionable or lower actionable conditions. On the right side of age 18, you have adult onset medically actionable conditions, which was again part of our parental decision-making study, and then adult onset non-medically actionable conditions, which we did not return in this particular study. So just to kind of describe the cohort, we had our affected cohorts with conditions such as PKU and MCAD and hearing loss, and a healthy newborn cohort. All of those were analyzed for the upper left quadrant of that diagram I showed you, so the actionable conditions that, with onset in childhood. We did this analysis blinded to the cohort identity so that we wouldn't know whether the patient was from the metabolic or the hearing loss cohort or was just a healthy newborn, and this was to give us a test of how well our analysis would do at actually predicting disease. We then had a secondary analysis for the patients who were in the affected cohorts, where we then looked in light of their phenotype to see if we could identify a diagnostic result. So some of the early results, we had essentially all negative results for the healthy cohort, which is exactly what you would expect. These are very rare conditions, and we don't expect to find very many of them in the small numbers that we've looked at so far. The metabolic cohort, fortunately, had a very high screen positive rate, meaning that we were able to identify positive results for the PKU patients and MCAD patients and so forth. But there were still several false negatives, and part of that was due to variants of uncertain significance that wouldn't have met our threshold for calling out as a positive screen, or heterozygous pathogenic variants where we didn't find the second allele. And this points to some of the deficiencies of genetic testing for metabolic conditions, in the sense that the metabolic analytes themselves are much closer to the phenotype and therefore perhaps a better thing to screen for. Interestingly, we did have one female PKU patient who we also found to be a carrier of OTC, and this points to the fact that we will be finding secondary findings in this setting as well. The hearing loss cohort was a bit more challenging in the sense that we had a lower positive rate, and many more VUSs in the diagnostic category. And this is not necessarily surprising, given that many cases of congenital hearing loss are non-genetic. And so this will be something, as we go forward in trying to predict hearing loss in patients. Again, it may be that the hearing screens themselves will be a more effective tool. The additional genomic findings in the non-medically actionable categories, or at least in the non-pediatric categories, showed us a adult onset medically actionable finding for ovarian cancer risk, as well as a child who was in our hearing loss cohort that ended up having a diagnosis of Lowe syndrome. It's probably unrelated to their hearing loss, but they may well have been enrolled in our study in the hopes of actually establishing a diagnosis for their condition. Again, the NC NEXUS project was a large number of people I want to acknowledge, particularly the co-PI, Cynthia Powell. So just to wrap up in the last couple of slides, I want to talk about genomic sequencing as a screen. If we restrict our results to known pathogenic variants in which we'll have a greater than 99% specificity, we're going to sacrifice the detection of true positives because there will be patients with novel variants that we will not report. This is going to be especially true when there are conditions with a high proportion of rare or private disease-causing variants. However, if we include results with less than 99% specificity, we will have greater numbers of false positives, and that will lead to the greater downstream costs and consequences to those individuals to whom we return those findings. So I think there are several factors that might influence our tolerance for false positives. And those things might be, for example, what would be the consequences of missing a diagnosis? And there may be some conditions where, in order to avoid harms to patients, we might want to have a higher number of false positives. I think if there's a gold standard follow-up test that can be done to adjudicate the genetic variation, I think that would be a reason to return more variation that might be essentially false positives, but would be accessible. And another consideration is really what would be the recommended management plan for the patient. Is this something that's going to be a relatively minor impact on their life, or it would be something major, such as recommending to have a gastrectomy, for example. We also want to look at the economic impact and the societal consequences. And so I think for all of us, we need to think about how we can engage with our own communities to start to think about, from a genomic sequencing standpoint, how will we decide which conditions to include and how we're going to manage that. So who should have screening, when will it happen and where, what should be screened for, how will we obtain consent to do that, who should pay for the screening, who should be responsible for following up on it, will parents and physicians be overwhelmed, how should we be performing the screening, who's interpreting it, how will it be disclosed, what types of variants should be reported, how many false positives will be tolerated. All of these questions we need to address as we start to set up this process in our communities. I want to thank you for your attention and take any questions.